So we have hit noon, so I think we're going to go ahead and get started because we have 4 presentations to get through today. We may have more trickle in as we tend to be a slightly late team. But thank you so much for coming to our staff symposium. We're going to have 4 presentations today. They've each been given a 15-minute increment. We're hoping we'll have a little bit of time for questions, but we may just need to stay and hold after. I have booked the room for a little extra time, and I think our speakers would be happy to help if you have questions after. Alrighty, so I think we have Peter Fritsch starting us off. And here you go. And I got you a timer.
Thank you, thank you, thank you. Thank you. Does it fit in here? Okay, yeah. There we go. Does this work okay? Can you hear me? All right, perfect. Thanks a lot. All right, thanks for joining us today. I guess I'm starting things off. And today I have the pleasure of talking to you about the systematics and evolution of the native blueberries of the Hawaiian Islands. I guess it's better if I hold it. So this is a collaborative project for sure. This is folks from the University of Florida. So Anna Becker's dissertation was based on this work in part, and then Nico Selenise and Pamela Soltis were her advisors, and Andrew Kroll is a postdoc. And a lot of the funding came from National Science Foundation back when that was a thing, I guess, I don't know, and with other funding as well. So species richness of Hawaii, 14,000 native non-ocean species, over 90% endemism. There's about 1,400 vascular plants. So really tremendously rich. And, you know, of course, many of these are rare and endangered species. It's a living laboratory for the study of evolution. They're very remote islands, oceanic. There's lack of predators and competition. There's a common volcanic substrate, so that kind of reduces another parameter. Diverse habitats, though, from rainforests to deserts. Island syndromes such as flightlessness and woodiness and selfing and gigantism and other things. And many of the groups have undergone what we call adaptive radiation, where it's like explosive speciation. And I just thought I'd throw in the image of Sherwin Carlquist studying the Hawaiian silver swords. Of course, we've got an NSF project right now digitizing Dr. Carlquist's collections materials. And of course, he was spent much of his career on studying the flora of Hawaii. Geologic history of the Hawaiian Islands is pretty straightforward. It's sitting on a hot spot, and so that hot spot creates volcanoes rising out of the ocean from time to time. And so it's a linear sequence because the Pacific plate is riding over this hot spot in a northwesterly direction. And so then you get the oldest island of Kauai over to the west and the youngest island of Hawaii, which is still building, over to the east. And this has given rise to what we call in biogeography the progression rule. The idea is that organisms jump from island to island as they're formed, and they can't easily go back because of something called niche preemption, which means that, you know, once those niches are filled on the oldest island, it's very difficult for a lineage to go back and fill, there's no unfilled niches left. So I'll come back to that in a bit. Okay, that brings us to Hawaiian blueberries. In Hawaiian, they're called ohelo, and they're part of the genus Vaccinium. They're typically montane shrubs. They occur in very rainy situations usually, and very acidic soils, in sphagnum bog, high elevations. But sometimes, as I showed you in the first image, sometimes they occur on bare lava flows, like on the island of Hawaii, where really almost no other plants can occur. Thought to be 3 species, each spread across each of the major islands. That's from the manual. I'll revisit that in a minute too. Well, they are called blueberries, I guess, by us, but they're really not blue. Most of them are red. And so there's more like a cranberry, which is also in the genus Vaccinium, than blue. Although there are some blueberries or blackish purple blueberries on the young island of Hawaii, and some orange ones too. Yeah, they are. Yeah. Mhm. They're in the section Myrtillus, which goes by the name bilberries. And those are all of north temperate regions across the world. They share, for example, the character you can see here, both of them, like the Cespitosum novalifolium are mainland species. They have single flowers in the axils of leaves. And they're also, the flowers are borne on current branchlets, not last year's branchlets and inflorescences. So that's unique to this section. But then they have their own set of characters as well. For example, most blueberries in the store, have a crown, right? But bilberries don't have a crown. Well, the mainland ones don't have a crown on them. You can see there's just a ring on the top of the fruit. But in ohalos, they all have a well-developed calyx. Again, it's kind of like a reversal back to the crown. And that has led people to think that this is a pretty old group if it's developing characters like that after it gets onto the islands. So we asked these following questions, and in the context of well, no one's addressed these questions before in Vaccinium of Hawaii. So this is kind of the first attempt to really do this. When, how many times, and from where did ohalo originate? Does it follow the progression rule? Did it spread anywhere else after it got to Hawaii? And how many species are there really? Okay, so phylogenetics, a little complicated here. Apologies. Don't read that, obviously you can't, but that's just intended to wow, okay, we sample a lot. This is the Trivaccinii, so we have to use the Trivaccinii to figure out where ohalo goes in the phylogeny. This is a nuclear phylogeny of 355 genes of nuclear sequence. Okay, so this is showing that the section Myrtillus is monophyletic, and embedded in it is monophyletic, which is great, ohalo group. So it looks like it dispersed to the Hawaiian Islands a single time. And also notable is this species Vaccinium cerium, which is from Southeast Polynesia. And so it looks like, well, three of the samples group outside that clade, but one group's within the clade. And now let's look at plastid phylogeny. Oh, I'm sorry, there's one more, Vaccinium ovalifolium to take note of. That actually groups within Myrtillus, but outside of the ohalo clade. Okay, now let's look at plastid. Similar kind of deal. It's lots of sequence data, but again we get a monophyletic Myrtillus, and I'm only showing in this case monophyletic ohalo group. In this case, the 3 or 4 samples of cerium all group within the ohalo clade, and so does one of our ovalifolium samples, which is pretty weird. But so we're interpreting this actually as hybridization. So once it dispersed to Hawaii, then dispersed to Southeast Polynesia, whereby it hybridized with a Southeast Asian species that occurs in those islands down there. And so pretty much nowadays is what you get as the hybrid there. The ovalifolium was very surprising because if it holds up, it'll probably be the either first or second, depending on how you interpret the data of a back dispersal to a continental area from Hawaii. It's very, very strange. We're hedging on that because we're a little worried about it. So we'd really like a lot more data to really confirm that. Okay, so back to kind of like biogeographic analysis per se, we used TreePull, which is a technical program to, estimate divergence times. There's no reliable fossils for Vaccinium, unfortunately. They're berries, so they don't preserve well on the fossil record. But there are fruits of adjacent tribes in the family, Eric, in the subfamily of Vaccinioideae. And so we used those. And so we were able to calibrate the tree based on those fossils. Now we use this program, BioGeoBears, was pretty standard. Okay, so timing and geographic origin, here's the kind of output you get with the areas are colors. And I'll just summarize for you without going into too much detail. Basically, dispersal from Japan or Northeast Asia about 5 to 7 million years ago. You can see the sister of the ohalo clade, which is blue, is Japan. And that's pretty cool because only 4% of those clades that have been looked at so far in Hawaii are from Northeast Asia. They're usually from the Southwest in the South Pacific, those tropical islands, and most of the others are from North America. So this is a very unusual pattern. The first emerging clade within Hawaii is Kauai. Well, that makes sense if it's an old lineage, and so as soon as Kauai popped out of the ocean, it took its opportunity and landed there. How did it land there? Birds, probably, yeah. Vaccinium cerium dispersed from Hawaii to Southeast Polynesia about 3 million years ago. So we had to throw out the hybrids because they won't work well with analysis like this. But there was this one sample that occurred in both plastid and nuclear. We think this is probably still a hybrid, but it's cooperating enough that we could keep it in the analysis and get an age estimate for it. And that's consistent with Southeast Asia biogeography, Southeast Polynesia biogeography. Here's Vaccinium yatabii, very cool, very little shrub, very rare in montane areas in Japan. It's got these zigzag stems and this yellowish green flowers. And lo and behold, what color is the fruit? Yeah, it's great. So at least it's consistent that way with what we think is probably the original fruit color. This is now published, just out a couple of weeks, and we're already getting some decent airtime. Thanks to our Marcoms group. And I was interviewed with an interview from Hawaii Public Radio. I hope that's going to be out maybe a week or so. We'll see. We'll see if I made the cut. All right. So now within Hawaii, we also did a study within Hawaii, phylogenetics and biogeography OHALA. Within Hawaii, we used 153 samples. We got something called single nucleotide polymorphisms, or SNPs, a lot of data from that. Again, big phylogeny. Don't worry about it too much. Just focus on the colors. The colors are the different islands within Hawaii, and you can see the pattern right away. I mean, you can see that they're blocks of colors. They're not scattered all over everywhere. So there's clearly some limitation to dispersal, and there seems to be strong island signal in this data. There's also some zigzags. Those are two different kinds of analyses. Don't worry about that so much, but we think the one with the strong diagonal is probably a hybrid species. All right, we're doing the same kind of biogeographic analysis and across the Hawaiian Islands, Kauai to Maui. Well, what's wrong with that? It skips over Oahu. So this is with just a straight analysis, and then there's even some back dispersals here, Maui to Oahu, and I think there's another back dispersal back to Kauai. Okay, well, that's all right. So we get that back dispersals. This thing probably gets around. You can deal with that. Maybe the niche preemption idea isn't so cool. However, there's a problem. And the problem is that is right here, where at the 4 million year mark, it's already dispersed to Maui, but Maui didn't exist 4 million years ago. So how do you deal with that? Well, you can do something called time slices. So you can constrain the analysis, and you can say, do not allow any dispersal to Maui or before 4 million years ago, or 2 million years ago, and the same thing for the big island of Hawaii. And when you do that, look what happens. It follows the progression rule almost perfectly. Yes, there's multiple from, say, Maui to Hawaii, but it's always a forward dispersal. And I don't think there's a single back dispersal in this whole analysis. So, that makes more sense in a way with the progression rule. But it does require another kind of assumption of, that we, ideally wouldn't use. But we just show both analyses, and, we have to just, say, this is what happens with the constrained, and this is what happens with the unconstrained. Finally, a bit about morphological diversity. Through Anna Becker's field work, she did all the field work for this study. You can see the kind of diversity we're dealing with, actually, and it's a lot more than your 3 species kind of idea. So we think there are probably 15 species based on the molecular study corroborated by the morphology. A lot of these actually were already named in the 30s by a botanist named Carl Scottsberg, who did a lot of work in Hawaii, but they named them as forms or varieties, and we think they're species. There are probably 5 or so more that don't have a name yet. So maybe there's 20 total. And there's a lot more field work that's needed and herbarium study. A lot of these, for example, haven't been collected, like this one on INAT. I don't think this has been collected before. Look where it grows. It grows right at the top of Kauai. It's not like people get there every day, so maybe it's not surprising that there are still undescribed things. And this one was in our study, and it's almost like a tree. It has no name, and it has no name for a reason. People have collected it before, but Scottsburg screwed up because he actually misapplied another name to this thing. So he wanted to call it Vaccinium callicinum variety montanum. He didn't list the type for montanum in this. So he's got a description for this thing. He cites the Harold St. John specimen there, and it's clearly that species, but it's not the name that he's using. So it's got a description, but it doesn't have a name or a type. So it's pretty weird. And then there's a lot of typology problems that still need to be worked out. This is like Archibald Menzies from Vancouver's voyages. He was the first one to collect these things, Europeans on Hawaii back in the 1790s. British Museum used to paste multiple specimens onto a sheet, and they just scribbled things on the sheet, so it's really hard to interpret some of this stuff. They have 2 barcodes on the sheet, but there's really 3 specimens on the sheet, and 2 of them are types. The middle one's a type, and that's the Menzies collection. That's a type of dentatum, but there's one below it that is a Godeshode collection. That is, there's a scribbled label there. It says plate 69. Well, that's clearly indicating the original publication. So that's clearly a type. All that stuff has to be worked out, and it's really labor intensive. It's also really fun, especially if you're into history, because there's a lot of like documentation of the voyages, when they were there, what they did, and all that kind of stuff. So there's a lot of work to be done. So O'Hara colonized the Hawaiian Islands once from Northeast Asia 5 to 7 million years ago. It generally seems to follow the progression rule, either constrained or unconstrained analysis, with perhaps some back dispersal. Dispersed from Hawaii to Southeast Polynesia and perhaps back to the continental mainland. And that might be the first documented case of that. And then the old age of O'Hara would predict high diversification, and the number of species is probably underestimated by as much as 6. So there's a lot of work to be done. And that obviously has implications for conservation. That's all I've got. So I'll take any questions if there's time. Looks like there isn't. Selfing is when a plant can cross with itself. So you have flowers, that either have the sexes in the same flower and then they can cross, or separate flowers, but they can still cross with themselves. Yeah. Any quick one? Quick questions? No, probably no time.
Yes, same. I think if we can hold them to the end, we're going to roll right into them.
Hi. Hello. Good afternoon. Thanks for coming today. I'm going to share with you information about my doctoral project. This is Hypotrachyna in continental Ecuador, morphological, chemical, and phylogenetic analysis, preliminary results. My advisors, Graciela Paz Bermudez from University of Vigo in Spain, and Manuela Dalforno Brit. I'm currently a PhD student of the University of Vigo in Spain. Okay, thank you. But first, who is Alba? Where is she from? I am from Ecuador. I lived in Quito. I work in the Universidad Central of Ecuador, where I teach ecology at a tourism student. And my lichenology life beginning with Manu here. in 2007, more or less. Then I took some courses about lichens, and I worked in Galapagos Islands too with lichens. And I stopped for some years, but now I decided to return and study lichens. So what is a lichen? A lichen is a symbiotic association between a fungus and an algae and or a cyanobacteria. You can see here sorry, I forget you can see here under a very powerful microscopy the fungus and algae. In the world, there are about 20,000 species and 1,002 genera and 119 fungal families. The lichens are important for environmental and ecosystem balance, pioneer species that contribute to soil formation, help to conserve moisture, and serve as indicator of environmental quality. Here, you can see lichens with diverse shapes and colors. This is just a small sample of their diversity. My main interest is the genus Hypotrachyna. Hypotrachyna is one of the most representative groups of the family Parmeliaceae, with 188 species, 100 of which are cited from Ecuador. Molecular analysis suggests it is polyphyletic. The genus is cosmopolitan, but it has its speciation center in South America at elevations 2,000 to 3,000 meters. The genus is characterized by having a narrow sublinear to sub-irregular lobes, truncate apices, and dichotomously branched resines. In Ecuador, this genus is very diverse. For this reason, I propose to analyze morphologically, chemically, and phylogenetic the species of Hypotrachyna from continental Ecuador. My specific objectives are to carry out an intensive survey of Hypotrachyna lichen species, perform morphological and chemical analysis of individual found, to analyze phylogenetically the species for their respective systematic and taxonomic delimitation, and implement socialization tools for the rapid identification of species in situ. But how do I do my investigation? I started with a field work in diverse ecosystems where I collected samples in different substrates in different altitudes. Here, there's some photos of the ecosystems. For the laboratory part, I worked in 2 labs. in the Universidad of Vigo, Campus Pontevedra in Spain with Graciela, and here breed with Manu. In these labs, I have analyzed the samples collected morphologically and chemically. Morphology, observing important taxonomic characters such as Isidia and Soredia, which are lichen propagulus, and chemically through applying tests to identify lichen substance. Also, I performed thin layer chromatography. It's beautiful, no? Too. To help identify a lichen substance and then its species. And also I did phylogenetic and molecular analysis following the different established protocols, DNA extraction, PCR amplifications, and sequences. Preliminary results, I collected 640 specimens in different substrates, and additional, I review material from different herbarium. During my work here in breed, I extracted 456 specimens and amplified all these new sequences. Next, I'm going to present our three from IT's nuclear marker barcoding for fungus. In this analysis, I included 262 new sequences plus 343 from gene bank for a total of 670 taxas. You can see here two clades, Hypotrachyna clade and Remotrachyna clade. Remotrachyna is a genus described recently. It's a sister group of Hypotrachyna. Some species treated as Hypotrachyna are part this clade now. For this reason, we decide to add this clade, Remotrachyna, to my project. As you can see here, this is the Remotrachyna clade proposed by Aybaca in 2010. And within, we found a new wealth supportant andin clade, which include these three species, Hypotrachyna verbidactylata with dactyls, Hypotrachyna sublevigata without propagules or no propagules, and Hypotrachyna brevirisa with Soredia. Historically, these propagules, they have been used for separate species. But these taxa don't present genetic differences and appear as single monophyletic species. Based on these results and the review phenotypic characters, we propose to combine and synonymize the three species into Remotrotrachina sublevigata. The article about this is coming. Now let's move to Hypotrachyna clade. A previous work reported 6 well-supported clade. And but our phylogenetic analysis now with a broader sampling corroborate with this and show 2 additional new clusters, mainly from the Andes. Finally, now I continue working, comparing species, writing. Thank you, breed family, all the persons here, especially Peter, Ale, Diana, especially for the los candidatos estudiantes, and Lucy, of course, Yvonne, Manu, of course. Thank you so much. I'm sorry for the bad English.
We actually have caught up now. So if we could hold till the end, that would be great. Just
Okay.
technically we're like 3 minutes under, but then.
Testing? Okay. Hello, everyone. I'm Kay Hankins, and thank you again for coming to attend this session today. I will be presenting our work here at the Botanical Research Institute of Texas and the Fort Worth Botanic Garden on accelerating Texas state conservation and habitat threat assessment using deep learning. So I want to provide a little bit of background and objectives about this project. Why we're doing this? Well, evaluating extinction risk and identifying the threats primarily driving that risk is vital to informing species conservation status assessment, management, and planning. However, producing these new status assessments or reevaluating those that are presently outdated is both time-consuming and resource-intensive. And just here in Texas alone, and this is probably an outdated number now, there are 437 ranked species of greatest conservation need, or SGCNs, with many more species awaiting formal status assessment or reevaluation all the time. So that presents a pretty major challenge in that not only the magnitude of need significantly outpaces what conservation practitioners can reasonably provide, but also so is the burden of proof necessary. That need is great to justify the severity of threats impacting target species populations. And that ultimately results in this sort of conservation status assessment bottleneck. And before I kind of get too far into kind of our further objectives and goals with this project, I kind of want to back up a bit and talk about conservation ranking and what all of that means. So conservation ranking here at the national level is following the NatureServe ranking schema and ranking calculator, which ultimately enables practitioners to evaluate conservation status of species here at the subnational level, which results in an S rank. So think of subnational level as your state rankings. This information can then be used to inform conservation statuses at larger scales, such as at the national level, which results in an N rank, and the global level G rank as well. So you can see here listed, those rankings range from 1 to 5, with the lower the number being more imperiled and the higher the number being more secure. So as an example, this beautiful native microheterotrophic orchid, native to Texas, Bledia or Hexalectris warnockii is a G2S2, meaning that it is a globally and state imperiled plant species. So what factors inform ranking calculation? Well, ultimately, there are 10 factors used to assess conservation status, each of which fall within one or three categories from which this species rank is calculated, including rarity, threats, and trends. Factors of rarity account for 70% of a species conservation status score, whereas threat factors only account for 30%. Yet this is the category that demands most of our attention. So what's the deal with this? Why is this such a big deal? Well, threats are very numerous and not always easy to empirically quantify with numbers and data. Publicly available data sets are plentiful, but they are largely unconsolidated and oftentimes differ in terms of spatial scale and resolution. There's also a lack of consistency in how practitioners approach this process because they do so differently, and they're not always using the same data sets or justification or sources. And ultimately, all of these factors then work together to further impede the efficiency of species status assessment. So that brings us back to what kind of inspired this project. Our goals were to then compile publicly available environmental data and resources to develop a proof of concept model for the development of ultimately a web-based tool to help support conservation practitioners who are doing this work in the field. And ultimately, the goal then being to improve the overall pace and consistency of formal species status assessment, help prioritize conservation targets, identify threats and data deficient species, and also expedite state and national review processes for at-risk taxa. So for the methods section, in order to actually do this, we needed data and we needed a lot of it. So a lot of the early stages of this project were primarily data acquisition and just aggregation. And a lot of that, thanks to Ashley Bales, was working with herbarium voucher specimens and records, georeferencing, getting kind of our occurrence data together for our data set, but also a lot of geospatial layers representing different threat variables as well. So what species did we use and why? Well, we started out with a small kind of arbitrarily designated sample of 50 species of greatest conservation need. These are species that are ranked S1 through S3 from TPWD's, the Texas Parks and Wildlife Texas Natural Diversity Database list. All were species that our team either had personal experience with or were in close geographic proximity to our respective institutions, which enabled easier ground truthing efforts. However, of these 50 species, only 44 had sufficient data to train models, some of which you see pictured here. But not only did we need sensitive species in order to train our models on what is a rare species, we needed to balance those models with an identical equivalent sample of more common and secure species presently ranked S4 and S5. So our more secure species. And that was, again, to balance out our model training set in terms of number of species selected, but also in the number of occurrence records for both of those binary categories. So for our threat variables, we identified and compiled as many geospatial data sets representative of each of the united classification of direct threat categories and their subclasses as possible. And this is what informs, this is what NatureServe uses to inform the threats in their ranking calculator. And broadly speaking, these threats fall into one of three major categories. Climatic threats, we've got your environmental threats, things like eco-regional, variation in terms of plant communities and habitats, soil, geography, these sorts of things, and also anthropogenic variables like land use. So in total, our data set consisted of 8,750 species occurrence records and 23 environmental and habitat threat variables, all of which we have compiled and downloaded and have represented it in a large kind of currently a Google Sheet. But this Google Sheet basically serves as a repository for historical data that, due to the current political climate, may not be as easily accessible. So we have these kind of backed up, and hopefully we'll be able to make these available to other practitioners interested in using these data sets in the future, including categories like what the data set is, a description, when it was published, when we downloaded it, how often it's updated, what NatureServe kind of threat category it's affiliated with, all of these things. But furthermore, aside from that, we needed to kind of classify our 8,750 species occurrences into kind of different response variable categories. So for our purposes, models perform much more reliably when you use binary categories of classification. So for us, generating predictions, models perform better when the predictions are threatened and not threatened, right? Instead of discrete categorical predictions where we're trying to predict whether a species is S1, S2, S3, S4, so on. So that's what we did. So we identified and associated all species with a conservation status S1 through S3 and classified it with the response variable as 1, which equates to threatened. And all those that were S4 and S5s received a response variable of 0, which meaning secure. And then all of these occurrence records were randomized by species into 5-fold training and testing data sets. So for model selection, we required pretty complex artificial intelligence models that were not only capable of handling diverse data sets, but also able to iteratively adapt through a process of trial and error to optimize the quality of model fit. So we chose gradient boosted machines and artificial neural networks. So what are these? Gradient boosted machines are composed of these decision trees that you see here. They use our environmental data to kind of partition our occurrences into those binary response categories 0 or 1. And for each successive decision tree, it explains less and less variation and is weighted accordingly when generating our model predictions. Artificial neural networks, on the other hand, are modeled after the human brain and work to analyze and process the data via a network of interconnected neurons or hidden layers that you see here in the middle. These neurons function to kind of identify and learn complex patterns and interactions in the input data that ultimately result in our model's predictions. So this is some of our kind of preliminary results. The resulting model predictions that we were able to generate via both of these different modeling methods, both gradient boosted machines and artificial neural networks, were highly similar, as illustrated in the image here on the bottom left. And although, even though that they were highly similar, GBMs, our gradient boosted machines, slightly outperformed our artificial neural networks, as evidenced by a Welch's t-test and a metric called area under the receiving operating characteristic curve. However, I will point out that while both of the results we have here are significant, the AUC scores listed here for both our gradient boosted machine models and our artificial neural networks, not the best. So AUC scores range from 0 to 1, with AUC of 0.5 being no better than random. So our models perform better than random, but there's still room for improvement. So where our models struggle. So if we're thinking about a kind of a confusion matrix where we've got our true positives, true negatives, what we're looking at right now are our false negatives. So these are species that are currently ranked as SGCNs that the models predicted were secure when they're in fact more threatened, right? So that's a pretty not, insignificant list of species there. So why is that? Well, at present, our data fails to account clearly for small and inconsistent population sizes. That's characteristic of parasitic plants and highly specialized microheterotrophic symbionts like Hexalectris mornacii here on the left, Cuscuta attenuata and Cuscuta exaltata. This is potentially because these species are highly specific to certain hosts or fungal partners. The known populations may not flower or put up inflorescences every year, and the data is sparse, as these species are likely very underreported. Furthermore, the data also fails to account for species we modeled with restricted range sizes, such as Calaroe scabriescula here at the left, Dahlia reversionii, Hymenopapus carizoanus, and Streptanthus bracteatus. But there was an interesting case, and I don't know if you caught it, but in my list earlier, there was an asterisk next to Yucca necopina. This is the Glen Rose or Brazos River yucca. The models classified it as a false negative, but is it really? This species is presently ranked as a G3S3, i.e. vulnerable. However, this species is now thought to be way more common than was originally believed. And so perhaps in this instance, our model's resulting prediction is not the false negative that it appears to be after all, indicating that perhaps this species is a good candidate for further review. On the opposite side of that spectrum, we have this list of species here. These are our false positives. These are common species that our model classified as being more rare. These are species such as Asplenium platyneuron, Batricium virginianum, and Eriocolon decangulare, which are more common, but are likely underreported. These tend to be ferns or your kind of less showy flowering plant species. So that likely has a lot to do with it. Whereas species such as Rexia virginica and Sarracenia alata are common, but in areas possessing a pretty high threat potential. And I'll circle back. You may have noticed another asterisk. Penstemon marianus, or the scarlet beard tongue, false positive, or is it? Spoiler alert. Probably this is another instance where our models actually correctly classified this species as being more rare than its ranking previously suggested. Because at the time we were assembling this data set, I was using a list of Texas species of greatest conservation need that did not include this species, but however, as we have since learned, an updated list exists, and that this species may now be considered an S3 species here in Texas. So perhaps not a false positive after all, and maybe this species is actually more vulnerable than its previous ranking suggests. So future directions and final remarks. Is this a tool at all intended to replace boots on the ground survey effort, population monitoring, or habitat threat assessment in any way? No, absolutely not. Just kind of think of it as another tool in the toolbox. Moving forward, we hope to expand our data set to include more Texas native plant species, threats, predictor variables, and factors such as date of original description and various organismal life history traits that also pretty significantly contribute to species rarity. So with that, just a big thank you to Anna Strong at Texas Parks and Wildlife and Texas Parks and Wildlife as a whole for providing the funding for this project. We're still ongoing, chugging along due to our collaborators, Chris Randall at SHSU, and a lot of really amazing undergraduates in that lab, Amy Gonzalez, Anna Maloney, Travis Rhodes, who is a graduate student, Marino Salazar, and Gabe Ramos, and lastly, Zyra Johnson. Lovely. This is them at TPCC, the Texas Plant Conservation Conference, Sam Houston State University, us at the Garden, Torch, Symbiota, Ashley, Brooke, everyone. Thank you so much for all of your time and input and advice. Thank you very much.
Hello, thank you. Today I'm gonna talk about ferns, which are super nice plants. And this is the result of one of the chapters of my thesis. I'm gonna talk about the phylogeny and character evolution of one really important clade in Elaphoglossum, that is the Lepidoglossa section. And this is basically gonna be the story of our approach to one of the most challenging fern genera. So I'm talking about Elaphoglossum, that is a very good representative of the tropical fern diversity. If you see in this map, it is spread all over the tropics. It has more than 600 species tropical distributed, and 75% of them are restricted to here, to tropical America. Elaphoglossum comes from the Latin words elapho means deer and glossum means tongue. So these are the deer tongue ferns because of their characteristic morphology of having these simple leaves. And another characteristic is that, and this is super interesting because normally when we think about ferns, we think about these very divided leaves, but these are also ferns. They are also characterized by having this dimorphism, that is basically the difference between the fertile leaf and the sterile leaf. That is the one that are gonna do photosynthesis only. This is a genus that has very large number of species and are very similar to each other. This is a very remarkable uniform genus. And the thing is, Elaphoglossum has been considered a challenge because most of its taxonomy is based on very subtle characters. One examples of these characters is these very tiny dots that we found over the leaves and over the rhizomes, that if we zoom it, they will look something like this. And these scales has super important characters that help us identify species. So because this group is so big, in order to make it more manageable taxonomically and have a better understanding of the group, what we have done historically is try to split the genus in several different subgroups. One of these infrageneric classification was made by Mikkel and Tortua in 1980. And these people divided the genus in 9 subsections and 9 sections and 29 subsections. So this classification was based only on morphology, but this time we use characters like, for example, the scales over the leaves. We also use the presence, they also use the presence of hydatodes, that are these pores in the margin of the leaves and in the end of the veins. Then they also use characters like, for example, this darkness and thickness in the base of the petioles. That's something that we call phylopodia. And they also use characters related with spore morphology. This classification was based only on morphological evidence. Later, with all the boom of the genetic data, we started to use more phylogenetic frameworks to find these groupings that will help us understand the genus. And here right now is the classification that we use in Elaphoglossum. This is a classification based on chloroplast markers, and what studies have found is that these 9 subsections, these nine subsections that were proposed on morphology, only 5 of them are monophyletic, and this is the groups that we have right now, that is basically 8 clades. And one of these 8 clades is Lepidoglossa, that is the clade that I'm working with. So Elaphoglossum section Lepidoglossa is a super interesting section in the genus. It has more than, based on morphology, we know that it has more than around 2,000 species distributed, also tropically. And here are some pictures of how the morphological variation that we can find within this clade. Species of this section are characterized by having these very, very scaly leaves, and that's why we find the name of the section, that is Lepido. In Greek means scale, so we are talking about the very scaly ferns here. And another characteristic of these ferns is having the presence of phylopodia that I just showed you, is there is this darkness in the petiole. But something really important about this section is these scales. What is very characteristic about these scales is that they have these unicellular marginal projections that are this synapomorphic character, a character that is unique for this group of species. Previous phylogenies have only sampled less than 30% of the species of this section, so we are still lacking a lot of in-depth evolutionary studies and a clear understanding of these morphological characters that will help us to define subclades within Lepidoglossa. And Elaphoglossum is a huge group. We saw that it has very similar species, but it's super interesting because it is a living example of rapid diversification in tropical regions. So by tracing its evolution, we can understand how and why this vast biodiversity have arisen in the tropics. And this is a reasoning behind our approach. We wanted to understand one of these challenging genera by studying its large section. So I have these specific objectives in my research. The first one is to generate a more complete phylogenetic approach for this section Lepidoglossa. In this case, we're using 3 chloroplast markers. Then we focus on testing if these subsections that were proposed on the base of morphology are monophyletic or not, meaning that if they form groups with a shared ancestor. And then we also wanted to identify these monophyletic groups that will help us to facilitate their later taxonomic revision. Our last objective was to investigate the character evolution of some important characters that have been useful for the classification of these species. How we did this, I'm not gonna go really deep into methodology, but the idea here is that we amplified these 3 chloroplast markers, and then we did this for more than 480 samples, which is a lot of sampling. This means an increase of around like 75% of, no, it means like a sampling that represents around 75% of the sectional Lepidoglossa, which is a huge increase. Basically what we did was just some analysis to obtain the most likely tree that have given risk to the data that we are seeing right now. And then at the end, I'm gonna show some results about some character optimization that is basically reconstructing the history of the characters. The taxonomic sampling consisted of around 75% of sectional Lepidoglossa, which is at least 93 more species than any previous study. So we consider this the most comprehensive phylogenetic reconstruction to date for this section. And interestingly, 23% of the species that are here represented in these warmer colors are from the Eastern Hemisphere. So this was a huge effort trying to gather all the information from both the Eastern and the Western Hemisphere. I'm going to show you now some preliminary results, and then by the end of my presentation, I'm going to show you an interesting story of a good finding that we had. So this is the beautiful maximum likelihood phylogeny. Here is very summarized for your better understanding. And what we are seeing here is that we recovered these 13 major clades that are here represented by the 13 color triangles, and in general are very well supported. How do we know the support? If you see the numbers right here, the higher the number, the better the support, basically. So we have very high support clades, and all these clades, what is seen super interesting is that all of them can be defined by morphology. We are using several characters to define these clades, like for example, rhizome architecture, the scale morphology. We are also using some characters that are, for example, are called resinous that I'm going to talk about later, but we are also using some characters related with spore morphology. Although we have these very well supported clades, we still lack of resolution of the relationships between these clades. We see, for example, different polytomies, that is basically where parts of the tree when we don't have a resolution of who is the sister of who. Although we don't have this resolution, we still have very well supported clades. And in fact, 3 of these clades have been studied before. So the idea is that we're still lacking the study of this other 10 rest clades that can be useful for later for future students to future students to go into the taxonomy of these clades. Other of my objectives was to infer if these subsections that were based in morphology are monophyletic or not. So if you remember, Mycolena tortua divided the genus, and within Lepidoglossa, they divided 6 subsections. What my data suggests is that only 2 of these subsections are monophyletic. How do we know? Because Here is the tree. We have mapped the different subsections in the tree. And like the idea is that we are looking for these clades, these groups that all the ancestor and all the descendants form a group. Only two of these groups are monophyletic, that is subsection muscosa here in red, and subsection petiolosa here in purple. The rest four groups are polyphyletic, meaning that they are spread all over the tree, and as we can see them, all the colors spread all over 11 different clades. And the reason why these subsection classifications are like this is because most of these classifications were based on either plesiomorphic characters that are basically share ancestral characters that are not very well informative, or convergent characters, meaning that they are the result of convergent independent evolution. One example of these characters that are convergent is these shiny and sticky dots that we find over the leaves and over the rhizomes that we call the resinous dots. So what our phylogeny suggests, and we see the tree in the bottom, is that the idea here, we try to reconstruct the history of this character along the phylogeny. And we're seeing that the ancestral state is having the absence of resinous dots. That's why we see these clades black. But then the derived character is the presence of resinous dots here represented in these clades in orange. What we are seeing is that this data suggests is that these characters has evolved at least 5 independent times along the phylogeny. That's why some of the classifications do not make sense with the evolution. Although we still use this character to define these well-supported clades, because the approach that we're using right now is trying to gather information from several morphological characters rather than only one. So we are using a combination of characters to define these clades. We also trace evolution of spore morphology. This was a huge and important work did by our undergrad research experience. From the NSF, Grace Pascarella, and she showed a beautiful poster in the botany conference the last summer. And basically her results showed that some of these characters in the spores are important and informative to define these groups. Here I'm showing you the evolution of these sculptural elements that we found in the spores. And what our data suggests is that, for example, spines are the ancestral character, and they have evolved independently different times into irregular deposits that are here represented in blue. Another character that is super important is the change of these sculptural elements into papilla. These papilla are a character that is unique for this clade, meaning that it is helpful for, it helps us define clades and understand different clades. Although the spines the irregular deposits are a derived convergent character because it has evolved more than once, it is still pretty useful for our taxonomic classifications to define clades. Now that we're talking about why morphology is super important in Elaphoglossum, I just wanted to share one last story that is called Things Are Not What They Seem. And this is basically the story of these species that Elaphoglossum rosenstocki, since the very beginning, this species has been placed in section Lepidoglossa because it has all the characteristics of any Lepidoglossa, very scaly leaves, and the scales that seems to have these unicellular marginal projections. Everyone thought that this was a Lepidoglossa species, so we did as well. And what happened is that in previous preliminary versions of our phylogeny, we found this sample nested within one of the sister clade that is the subulate species of these ferns. This section setosa is very different from Lepidoglossa. They are characterized by having these hair-like scales that are very different to the flat ones that we find in Lepidoglossa. So we were just thinking that it was matter of a contamination, and we were thinking to just get rid of this sample, and we try another one again. But this new sample again fell into the setosa clade. So we were wondering, okay, let's look in more detail what is going on here. And the first thing that we notice is that when we look at the scales in the microscope, they have the presence of these enrolled bases that are very similar to the ones that we find in the setosa clade. That was not enough evidence because this character has been reported before in Lepidoglossa, so we need to look more in detail. The other interesting morphological characters that we found is that when we examine the scales into 4EX microscopy, we notice that these marginal projections are not unicellular. And if you remember at the beginning of my talk, I told you that the characteristic of Lepidoglossa is having unicellular projections. And we are seeing here that these projections are made by 2 adjacent cells. So this was enough evidence to tell us, okay, this is not a Lepidoglossa species as we thought. Then some later evidence using SEM pictures from the spores, we noticed that the spores look like a normal, typical Setosa clade species. So we just needed molecular phylogeny, duplicate of samples, light microscopy, SEM pictures, and all of these to figure out that Elaphoglossum rosestockii is not a Lepidoglossa species, but is rather a Setosa species. So I just hope that not all the species in Elaphoglossum are as troublemaker at this one, but I know that they are. But this is a super interesting example of why we need morphology along with evolution and phylogenetics to understand the species, and that we cannot take these two approaches apart. So some future directions of my project is including new samples from different projects that are happening in Hawaii and the Philippines. Also, the idea is in the future to wonder more about the biogeography of this group of species, especially using nuclear markers, because we wonder if with the use of nuclear markers, we have more resolution and the one that we are having with plastid markers. And yeah, I just want to say thank you to all the people that have been involved. This is a huge project that has been running for a lot of time. So yeah, thank you.
So we are actually already after 1 o'clock, but I think our speakers are all willing to hang out. If you guys have any questions after, thank you so much for attending.